Hello, my name is Ed Chapman, and this video cast is going to cover the second part of membranes, and we're going to be focusing now on the function of transport. Passive transport requires no energy, and it's also known as facilitated diffusion. It's a way that cells use embedded proteins to provide a gateway or a channel through which solutes can diffuse down their concentration gradient into or out of the cell. It has the ability to control which molecules can pass through the gate, but it requires no energy, so we can say that it's diffusion driven. Here's a picture showing you passive transport. We have a carrier protein embedded in the phospholipid bilayer. We have some solute molecules concentrated on the outside of the cell in the environment. And as you can see, this carrier protein has the ability to change its shape and allow the solute molecules to diffuse inward into the cytoplasm, which is down their concentration gradient, which is free. It requires no energy. An example of passive transport are aquaporins. Aquaporins are proteins embedded in phospholipid bilayers that provide a channel through which water molecules can diffuse very quickly. So aquaporins make osmosis happen faster. Remember, water molecules can also get through the phospholipid bilayer, but that's a relatively slow process. The aquaporin system allows osmosis to happen even faster than it would without them. Active transport, as its name says, requires energy, and it involves these integral proteins and ATP energy to move molecules against their concentration gradient. So they're not going to be flowing down their concentration gradient, they're going to be flowing up it, so to speak. And this flow doesn't happen without some energy investment. This is a big deal because about 80% of the ATP that your cells require from moment to moment is going towards powering these active transport pumps. And the most important of all the active transport pumps is the sodium-potassium pump. And the sodium-potassium pump works in this way. It's going to be pumping sodium ions out 3 at a time, if you follow this diagram, and it's going to be pumping potassium ions in 2 at a time. And you'll notice as we move from picture to picture in this diagram, ATP is being used to power the shape change of this embedded protein. And depending on which way the protein's gate is open, sodiums can get in and out or potassiums can get in and out. But the net result of this process is 3 sodiums are being pumped out into the environment for every 2 potassium ions being pumped into the cytoplasm. So this is a pump because it's maintaining a gradient. It's forcing molecules in a direction they otherwise would not diffuse. Now, electrogenic pumps, like the sodium-potassium pump, generate an electrical charge across the cell membrane. And this is basically what batteries are. Electrogenic pumps can use living cells to generate a little bit of electricity. The sodium-potassium pump, as you probably noticed, is pumping 3 sodiums out for every 2 potassiums that get pumped in. This isn't a balanced movement, so we're accumulating positive charge on the outside of the cell membrane, and we're getting rid of positive charge on the inside. And the net result of that is we can generate some energy. Proton pumps are another type of pump that do the same thing that the sodium-potassium pump does, except instead of moving sodium ions and potassium ions, it pumps hydrogen ions. And both of these processes use ATP energy to function. Cotransport is a situation where a proton pump, which pumps hydrogen ions out of the cell, is combined with another embedded protein that's functioning as a facilitated diffuser for a molecule like sucrose. And in this case, one pump, the proton pump, is using ATP to move hydrogen ions out, and then these hydrogen ions are allowed to diffuse back in through a different protein, but as they diffuse back in, they're bringing along with them for the ride another molecule. In this case, it's sucrose. So in this case, the proton pump is a co-transporter of glucose, or in the case, well, that should say sucrose here. So in this case, it's transporting sucrose into the cytoplasm, and it's doing that with energy that's coming by way of the gradient created with hydrogen ions. Bulk transport is a form of transport where a cell actually changes its entire cell membrane shape to move materials inward or outward. And there are 2 directions. You can either move things out or into the cell. If you're moving things outward, we call that exocytosis. And if you're pulling things into the cell, we call that endocytosis. Now, endocytosis is a way that a cell can take in macromolecules and other particles into the cytoplasm from the environment. And there are three types of endocytosis: phagocytosis, pinocytosis, and something called receptor-mediated endocytosis. We'll talk about that last. Phagocytosis is also known as cell eating, and in this picture you can see a macrophage, which is a very large type of white blood cell, eating some bacteria. I believe these are tuberculosis bacteria, which are colored here in yellow, and they're being surrounded and engulfed by the cell membrane of the white blood cell, or the macrophage. And this is a great example of phagocytosis, or cell eating. Endocytosis, that takes in droplets of liquid dissolved in whatever the environment, the aqueous solution of the environment, is called pinocytosis, also known as cellular drinking. And here you can see how the cell membrane is changing shape to absorb a little droplet of material here and have it now in a food vacuole inside the cytoplasm. So altogether, phagocytosis, pinocytosis, and finally there's receptor-mediated endocytosis, which works by the cell membrane having special little proteins that can bond to certain things, certain substrates in the environment. And once they're all filled up, the cell membrane is then triggered to change shape, and these materials are now encapsulated inside of a food vacuole or a vesicle that has these special proteins around it and inside it. So these little receptors are able to bond or dock, and we get a enzyme-substrate reaction here, and that makes it a little bit more complicated than plain old phagocytosis or pinocytosis. All right, that's all that we have to do here, and I promise my video face will be back next time. It's too dark in this room to make the video with that running today.